Hello all, my name is Neha Pandit, Assistant Professor of the Dr. D.Y. Patil Institute of Pharmaceutical Science and Research. Today I am going to present the topic that is Basic Principles of the Polymerase Chain Reaction. This comes under the course outcome that is BP605T1. So what is that? It is the knows the basics of the biotechnology techniques and the various systems used in the biotechnology. So before starting the PCR, you should know the basics of the genetic engineering. So what is the best genetic engineering? It is a process of manipulating the genetic material of an organism. So what is genetic material? DNA and RNA, that is called as genetic material, by introducing, removing or modifying the specific DNA sequence to produce the desired characteristics or the new traits. Or another definition of genetic engineering is that it is the artificial manipulation or alteration of the gene that is called as genetic engineering. So genetic engineering in that recombinant DNA technology, that is rDNA technology, is the main pillar, and it is a technique to alter the genes of an organism. So what are the steps involved in the genetic engineering is that first isolation of the DNA, second cutting of the DNA, third amplification of the DNA, that is done with the help of the PCR, and fourth formation of DNA, and fifth gene transfer or the transformation. So in the next slide we will see the what is mean by PCR. So what is PCR? PCR is the laboratory technique. So laboratory, that is called as in vitro. If it is happens in the body, that is called as in vivo. So PCR is the in vitro process, and it is used for the generating the large quantities of the specified DNA. It helps to synthesize the multiple identical copies of the DNA of interest, and it was first discovered by the Carey Mullis in 1983, and it was a Nobel laureate in 1993. So nowadays PCR is a basic tool for the molecular biologies. So what is the principle behind the PCR? So in this PCR there are three steps involved in that. First is the denaturation, second is the renaturation and or annealing, and third is the synthesis or the extension. So in the first process, the double stranded DNA molecule is denatured into the two separate strands. In the second step, each strand is then allowed to hybridize with a primer, that is called as renaturation or annealing process, and then the primer duplex is formed for DNA synthesis with the help of enzyme DNA polymerase. So these three steps, that is denaturation, renaturation and synthesis, are repeated again and again to generate the multiple copies of the DNA. So in this slide, we will see the what are the essential requirements of the PCR. So first, a targeted DNA having 100 to 35,000 base pair in the length, two primer, that is the forward primer and the reverse primer, which are short nucleotide in the length, that is 17 to 30 nucleotides, and that are complementary to the targeted DNA. Next is the 4 deoxyribonucleotides, that is the DATP, DCTP, DGTP and DTTP. So what is ACGT? Means A means adenine, C means cytosine, G means guanine and T means thymine. So next last is the DNA polymerase. So that is the Taq polymerase that can withstand at the higher temperature up to 95 degrees Celsius. That is the thermostable Taq DNA polymerase. So the reaction mixture contains the target DNA, two primer, thermostable DNA polymerase, that is the isolated from the bacterium Thermus aquaticus, that is stacked DNA polymerase, and the 4-deoxyribonucleotides. So in the PCR, each cycle of the PCR contains the three stages. So there are, first is the denaturation, second is the renaturation or annealing, and third is the extension. So in the denaturation process, there is a temperature, it is about 95 degrees Celsius for the 1 minute. So double stranded DNA, it gets separated into the single strand. And in the next step, that is second, that renaturation or annealing, in this the temperature slowly cools down to up to 55 degrees Celsius, and the primer base pair with the complementary regions that flanking the target DNA strand. So this process is called as renaturation or the annealing. So high concentration of primer that ensures the annealing between the each DNA strand and the primer rather than the two strand of the DNA. So in the third step of the PCR, that is synthesis or extension. So in this step, the initiation of the DNA synthesis occurs at 3' hydroxyl end of the each primer, and the primer are extended by joining the bases to the complementary to the DNA strand. So however, the temperature has to be kept optimal that is required for the DNA polymerase. So the temperature required for the tag DNA polymerase is the 75 degrees Celsius. So when PCR was find out that time E. coli DNA polymerase was used. So the temperature required, optimal temperature required for the E. coli DNA polymerase is the around 37 degrees Celsius. But this in this third step of the PCR, the temperature goes around 72 degrees Celsius. So that's why this enzyme not able to withstand that temperature. So the in nowadays tag DNA polymerase is used for the in the third step, that is synthesis step of the PCR. So this reaction can be stopped by raising the temperature to about 95 degrees Celsius, and the three stages of the PCR in relation to the temperature and time, the PCR takes the 3 to 5 minutes of the each cycle. So in the normal factories, the PCR is carried out in an automated machine. So the new DNA strand joined to each primer is beyond the sequence that is complementary to the second primer. So these new strands are referred as a long template and they will be used in the second cycle. And for the second cycle of the PCR, the DNA strand, that is the original plus newly synthesized long template, are the denatured anneal with the primer and subjected to the DNA synthesis. So at the end of the second round, long template and short template DNA strand with the primer sequence at one end and sequence complementary to the other end primer are formed. So in the third cycle of the PCR, the original DNA strand along with the long and short templates are the starting material. So techniques of the denaturation, renaturation and synthesis are repeated again and again. So this procedure is repeated and it is estimated that at the end of the 32 cycle of the PCR, about million fold of the target DNA is synthesized. So in this slide, you will see the three cycles of the PCR, the three stages of the PCR in one cycle, as well as the PCR components. So what are the PCR components? They require the DNA sample, primer, nucleotides, tag polymerase, mixed buffer and PCR tubes. And in each cycle, you can see here, the first step is the denaturation. It is about 95 degrees Celsius. Second stage is the annealing. It is around 55 degrees Celsius, and third step is the extension. It is around 72 degrees Celsius. In the first step, double stranded DNA, it gets separate out into single strand, and in the second step, annealing, in that forward and reverse primers are attached to the complementary strand, and in the third step, the newly synthesized DNA are formed. So this is the PCR process in the one cycle. This process happens again and again. So that's why they produces the million copies of the DNA, and this whole process occurs in the machine that is called a thermal cycler. So the next is the sources of the DNA polymerase. So in the original techniques of the PCR, Kenlo fragments of the E. coli DNA polymerase was used. But this enzyme gets denatured at the high temperature, therefore fresh enzyme had to be added for each cycle. That's why in the 1989, the lawyer, with the introduction of the tag DNA polymerase from the thermophilic bacterium, that is the Thermus aquaticus, the DNA polymerase is heat resistant and hence it is not necessary to freshly add this enzyme for each cycle of the PCR. So next is the what are the key factors for the optimal PCR. So first is the primer. It plays a significant role in the determining the PCR. Second, the primer should be short in length, nucleotide length, that is 17 to 30 nucleotides, without secondary structure and without complementary among themselves. So these are they themselves are ideal. So the complementary primers can hybridize to from the primer dimer and gets amplified in the PCR. So when we select the primer, it should be without the secondary structure as well as without complementary among themselves. So next key factor is the DNA polymerase. So tag DNA polymerase is the preferred as it can withstand at the high temperature. So therefore, in the hot start protocol, so DNA polymerase is added after the heat denaturation step of the first cycle. So this avoids the extension of the mismatch primers that usually occurs at the low end, low temperature. So in the third key factor is the targeted DNA. So in general, the shorter the DNA or sequence of the target DNA, the better is the efficiency of the PCR. So however, in the recent year, amplification of the DNA fragment up to 10 kb has been reported. So the sequence of the target DNA is also important in the PCR, and in that the GC means guanine and cytosine rich regions of the DNA strand are mainly selected for the targeted DNA. So next key factors are the promoters and the inhibitor. So in that, addition of the protein, that is the such as bovine serum albumin, that is BSA protein, that enhances the PCR by protecting the enzyme DNA polymerase, because the humic acid mainly found in the archaeological samples of the targeted DNA that inhibits the PCR. That's why we use the protein, that is the BSA, that is bovine serum albumin, to prevent the inhibition of the PCR. So next is the what are the application of the PCR nowadays. So in that, first, the PCR used in the clinical diagnosis. It is used for the various diagnosis of the diseases in the humans. So these include the diagnosis of the inherited disorder, that is the genetic diseases, viral diseases, as well as bacterial diseases. The occurrence of the genetic diseases frequently identified by the restriction fragment length, that is RFLP, that can be employed only when there is a mutation resulting in the detectable change in the length of the restriction fragment. So for all such disorder, PCR technique is a real boon, and it provides the direct information of the target DNA. So this is done by the amplification of the DNA of the relevant region, followed by direct analysis of the PCR product. Second is the prenatal diagnosis of the inherited disease. It helps to PCR is employed in the prenatal diagnosis of the inherited disease like sickle cell anemia, beta thalassemia, phenylketonuria can be detected by using the PCR. So third use of the PCR is that diagnosis of the retroviral infection. So it helps in the diagnosis of the and monitoring of the retroviral infection, that is the HIV infection. So next is the diagnosis of the bacterial infection. So bacteria like tuberculosis by Mycobacterium tuberculosis. The next use is the diagnosis of the cancer, mainly cervical cancer, that caused by the human papilloma virus can be detected by using the PCR. So next is the sex determination of the embryos. The sex of the human and the livestock embryos fertilize in vitro and can be determined by using the PCR. So further, this technique is also helpful to detect the sex-linked disorder in fertilized embryo. So in the next application, PCR in the DNA sequences, the technique is a much simpler and quicker to amplify the DNA, and it is conveniently used for the sequencing. So for this purpose, single strands of the DNA are required. So next, PCR in the gene manipulation and the expression studies. So the main advantages with the PCR is that the primers need not have the complementary sequence for the targeted DNA. That's why the sequence of the nucleotide in this piece of the gene, that is target DNA, can be manipulated and amplified by using the PCR. So PCR is important in the study of the mRNA, the products of the gene expression, and this is carried out by the reverse transcription PCR, that is the RT-PCR. So in the next use, that is a PCR in the comparative studies of the genome. So PCR nowadays help in the comparison between the two organisms that can be measured by PCR with a random primer. The products are separated by the electrophoresis for comparative identification, and PCR is a really very important in the study of the evolutionary biology, more specifically referred to as the phylogenetics. Next application is that it is used in the forensic science. So it helps to detect the criminals. The single molecules of the DNA from any source, that is the blood stain, hair, semen, etc., of an individual is adequate for the amplification by the PCR. That's why PCR is a very important for the identification of the criminal. And this reader may refer the DNA fingerprinting technique. So next application is that in a comparison with the gene cloning. So PCR has several advantages over the traditional gene cloning technique. So these include better efficiency, minute quantities of the starting material, that is DNA, cost effectiveness, and minimal technical skill and time factor. So in due course of the time, PCR may take over most of the application of the gene cloning. So these are the basics of the PCR, that is the polymerase chain reactions, I have explained from this slide. So in that you have seen the three stages of the PCR, that is denaturation, renaturation and the annealing, components of the PCR, key factors of the PCR, as well as amplification of the PCR. So thank you.